Hello guys, this is Vinith and welcome to the molecular biology series. So today I'll be discussing about the eukaryotic transposons and their transposition mechanisms. Okay. So if we talk about these transposons which are present in the eukaryotes, they are categorized into two different groups. So one is the DNA transposons and second group involves the retrotransposons. Okay. Retrotransposons. So these DNA transposons resemble the transposons which are found in the bacteria. Okay, so bacteria also contains a lot of DNA transposons. Okay, the same type of DNA transposons are also present in the eukaryotes. Okay, if you look at the examples of this DNA transposons, so one is the P element in the Drosophila. So these P elements which are present in Drosophila mainly responsible for the hybrid dysgenesis. Okay, so that means they make the sterile hybrid. Okay, and then we have the AC and DS elements which are found in the maize corn kernels. Okay, so these AC and DS elements are responsible for the production of different color patterns of these corn kernels. Okay, so different corn kernels will be seen in different will be having different types of colors. Okay, the purple color, the yellow color, the white color, or the transparent ones. Okay, sometimes the brown, green colors. Okay, so this kind of color, different color patterns, or this kind of different phenotypic variations will be produced by this AC and DS elements. Okay, because of presence of this kind of transposable elements in the corn kernels, these kind of different color patterns will be seen. Okay, then if we talk about the human genome, approximately 350,000 of DNA transposons are present in the human genome, and most of these DNA transposons are not active. Majority of them are not active because non-functional transposase gene, the gene which code for non-functional protein, transposase protein, and and it might be due to the mutated sequences which are present present at the terminal ends. Okay, so when these transposal elements contains some kind of mutated sequences in their terminals, the transposase enzyme will not act on these transposable elements and will not cut the sequences, so they cannot transpose into different locations. Okay, so because of presence of mutated sequences at the ends, or because of presence of non-functional transposon genes. Okay, so most of the transposed elements are inactive inside the human genomes. Okay, that is why human genome quantity is not increased during the course of evolution. Okay, only about 35 to 40% of the genome has increased. Okay, and that is mainly due to these transposons only. Okay, then what we have is the retrotransposons, okay, which use the RNA intermediate for their transposition activities. Okay, so what they do is first they undergo transcription and produce an RNA molecule. So this RNA molecule will again be converting into DNA molecule or the RNA DNA hybrid, so by using this reverse transcriptase enzyme. So this RNA DNA hybrid undergoes cleavage by using RNase H enzyme. When it gets separated from the RNA, this DNA will now get integrated into different locations of the chromosome and again get duplicated in that location. Okay, producing a retrotransposon in different location and conserving the retrotransposon on the original locations. Okay, so these retrotransposons are completely different from the DNA transposons. Okay, so DNA transposon majorly transpose via conservative mode of transposition mechanism, while the retrotransposons transpose via replicative mode of transposition mechanism. Okay, so this is what the difference between these two. And we have TY elements in yeast and copia elements in the Drosophila, and also ALU sequences in the humans. So these ALU belong to the short interspersed nuclear element categories, which doesn't contain any kind of long terminal repeats. Okay, the difference between this TY element and TY element, copia elements, and the ALU elements is these two are regarded as the LTR retrotransposons. Okay, LTR retrotransposons is a retrotransposon which contains long terminal repeats. Okay, repeats are very long. Okay, terminal repeats are very long, while the ALU elements, which belong to the short interspersed nuclear element category, science category. So this ALU element doesn't contain any kind of long terminal inverted repeats, but act as a retrotransposons. Okay, they also produce some kind of transposase enzymes. Okay, they actually don't produce transposase enzyme. They are completely dependent on the long interspersed nuclear element sequences. Okay, so lines will be producing the transposase enzyme, and this lines will be producing the reverse transcriptase enzyme. That this reverse transcriptase enzyme will activate the ALU elements inside the nuclear genome. Okay, So this is what the major roles of these transposable elements, or some major points of the transposable elements, okay, and the categorization, okay. So if we talk about the percentage of the transposable elements in the nuclear genome, okay, so majority of the transposable elements are found in the maize, okay. So maize actually contains about 85% of transposable elements in 85% of transposable elements in their nuclear genome, okay. That means their genome majority 85% of the genome is occupied by the transposable elements, okay. And the second, we have the frog, this one. So about 75% of frog's genome is occupied by the transposable elements, okay. And the least comes to the yeast, okay. So least one is the yeast. So it nearly has about around 6% to 7% of nuclear genome composed of transposable element, okay. Since this is a unicellular some kind of multicellular organisms, okay. So this is a yeast is a unicellular organism, okay. So that means only one cell is present, okay. So which also contain the minimal amount of the DNA. So that means there is no possibility of occurring possibility of transposable materials occurring various very major quantities, okay. So that is what we're seeing in the yeast, okay. Humans also contains a lot of transposable elements. Around 45 to 50% of the human genome contains this transposable elements. So I've said 350,000 transposable elements are present in the human nuclear genome, okay. So this is about the percentage values of transposable elements in the nuclear genome. Then we have different members of the transposable families, that they are the autonomous transposons and the non-autonomous transposons. These are the different types of members of the transposon families, okay. So the autonomous transposon members, okay, which belong to some kind of super transposon family of the transposable element. These autonomous transposons have the ability to excise and transport by themselves, okay. That means they can able to produce a transposase enzyme, and they can able to integrate themselves into different locations, okay. And they transpose via cut and paste transposition mechanisms, okay. They use the conservative mode, okay. While we have the second category as the non-autonomous transposons, okay. So these non-autonomous transposons, they actually move into different locations, okay, by utilizing the enzymes of non-autonomous transposons, okay. So these non-autonomous transposons are dependent on different types of intervening sequences, okay. So that means they require autonomous transposon for their activity, okay. Whatever the enzymes that are encoded by these autonomous transposons are utilized by this non-autonomous transposons, and by utilizing those enzymes, they will move into the different locations of the chromosomes, okay. These non-autonomous transposons actually do not encode for any kind of transposase enzymes or integrase enzymes, okay. They are completely dependent on the autonomous elements of this families of transposable elements, okay. Within super families, we will be having only single type of autonomous transposons, but variety of non-autonomous transposable elements, okay. That means if we talk about any super family, we'll be seeing only one element which has this autonomous property, which can encode for a transposase enzyme or integrase enzyme, okay. Only one single type will be there, and using this single type autonomous transposable element, lot of non-autonomous elements will move into the different locations, okay. So this is what about the different classes of this transposon members, okay. And if we talk about the mutator, if you talk about the maize, the major transposon elements in the maize are mutated transposons as well as the AZ and DS elements, okay. These mutated transposons contain autonomous element called as a MUDR element, okay. These are quite mutagenic in the maize plant, okay. So they cause a lot of mutations in the maize plant. So these mutated transposon contains an autonomous element called as a MUDR element, okay, which codes for different enzymes, okay, transposase enzyme and the integrase enzyme. It contains two different opening frames or the two different genes, okay. So this autonomous element contains two different genes, okay. Gene A, or you can also call that as a MUDRA. It codes for the transposase enzyme. This one, MUDRA gene codes for the transposase enzyme, and MUDRB, it codes for the enzyme which is responsible for the integration, okay. That is the integrase enzyme, okay. And these two genes which are present in this element, autonomous element, okay. So these are flanked by 200 base pair inverted repeats. Okay, that means if you consider this is a long DNA sequence, actually an autonomous element. Okay, so here what we have is the two operating frames, MUDRA and MUDRB. So these two genes are flanked by 200 base pair inverted repeats, which runs in opposite directions like this. Okay, here we have one inverted repeat, and here we have another inverted repeat. Okay, so these two inverted repeats will be oriented in opposite directions. Okay, and they are present in either sides of the gene A and gene B. Okay, and then we have the non-autonomous mutator elements, which are completely dependent on the enzymes of this autonomous element. Okay, by using those enzymes, it will get mobilized into different locations. Okay, now mutator element which are found in the maize plant. Okay, they are regarded as the founding members of this mu-like element superfamily of transposons. Okay, so there is a superfamily called as the mule superfamily or the mu-like element superfamily. So this mu-like element superfamily contains a lot of different transposons. Okay, autonomous and non-autonomous transposons. Okay, so in this superfamily, these mutator elements are considered as the founding members. Okay, the major members. Okay, and these are actually present in the bacteria, fungi, plants and animals. Okay, and these mu elements, they transpose to different locations. Okay, and the frequency of the transposition is approximately 50 to 100 copies per generation. Okay, for one generation, the frequency is about 550 to 100 copies. Okay, and if you want to transfer a mutated transposable element into different locations, okay, so that region should be in the open chromatin form. Okay, that is what I've given over here. Okay, whenever you want to insert a mu-like element or the mutator element, okay, in the genome, okay, so that genome should be in the open chromatin form or in the euchromatin form. Okay, so then only the insertion will be made possible. And when the region stays in the open chromatin form, this will also enhance the insertion rates and also enhance the recombination rates for this the mule elements. Okay, so this is what about the mutator family transposons. Okay, here we have the superfamily of this mule elements. Okay, or mule-like elements. Okay, so this superfamily contains lot of transposon elements, mu 1.4, mu 1.7, mu 3, mu 4 and mu 7 or RCY. And then we have this MUDR autonomous element, okay, which contains MUDR A gene and MUDR B gene. Okay, if you look at this MU4, it also contains a brassinosteroid incentive 1 gene. Okay, and this is mainly present in the Arabidopsis plant. Okay, so this plant contains this MU4 transposer elements, which mainly contain the brassinosteroid incentive 1 gene. Okay, and if you look at the actual structure of this mutated transposon, okay, so this autonomous transposon separate and non-autonomous transposon element is separated by some kind of intervening sequence or some kind of spacer region over here. So this spacer region separates this autonomous and non-autonomous transposon of the chromosome, and when this autonomous transposon transcribes, it produces the enzyme called as the transposase. Okay, and this has the ability to move independently to different location. Okay, independently it moves from one location to another location, but whatever the transposase that is produced from the transcription process of this autonomous element, okay, so this transposon will now be acting on the non-autonomous element for the mobilization of this non-autonomous element. Okay, so if the non-autonomous element want to get transposed into different location, it requires the enzyme of this autonomous element. Okay, the enzyme of this autonomous element. Okay, and then it moves into different sites. Okay, so this is about the mutated transposon, which majorly causes the mutagenesis, which majorly causes mutations in the maize plant. Okay, they are the major transposons responsible for the mutations. Okay, then what we have is the AC and DS elements, which were discovered by the Barbara McClintock. First, she called them as the controlling elements or the jumping genes. Okay, because the DS elements are the molecule which jump from the one location to another location on the chromosome frequently. So there was a scientist who first observed this color patterns or the mutation patterns on the in the nuclear genome of this corn kernels. Okay, and he actually forwarded his work to this Barbara McClintock, and this Barbara McClintock studied work of that scientist and said that and concluded that the mutations were mainly caused due to some chromosomal breakages. Okay, and these chromosomal breakages were mainly caused by the some kind of DNA sequences which will be jumping from one location to another location. Okay, by hearing this statement, a lot of scientists laughed at her and actually ignored her work. Okay, so her work was ignored for many years. Okay, and after actual discovery of the molecular techniques like the PCR, the RFLP techniques and all these things. Okay, so her work was recognized, and a Nobel Prize was also awarded for her work. Okay, the Barbara McClintock work. Okay, so she was discouraged by a lot of fellow scientists in her university. So that is another topic. But she was motivation for a lot of women to get into the science fields. Okay, so she was the one, and also we have one more major scientist, women scientist called Rosalind Franklin. Okay? Rosalind Franklin and the McClintock, they empowered a lot of women to get into the science. Okay, they to opt the science as their major subject. Okay, so this is about the Barbara and McClintock and the AC and DC elements. Okay, so these mutations, as she said, okay. What she said is, the mutations were majorly caused due to the chromosome breakages, and these chromosomal breakages were majorly caused by the jumping genes. Okay, so these jumping genes are actually called as a controlling elements. Okay, the controlling elements are what dissociation and activator elements. Okay, so these are expressed in the endosperm or alveolar layers of the corn kernel plant. Okay, so what she said is, based on her genetic observation and the cytological experiments, what she said is, there was a dissociation element that is present in the chromosome 9. Okay, and there was also an activator element which was also present in the chromosome 9, or sometimes it may not present in the chromosome 9. Okay, it is absent on the chromosome 9. Okay, but if the activator element, if it is present in the chromosome 9 along with the DS element, okay, so this AC element will cause the DS element to induce the breakage at a point on the chromosome and get transposed into different locations. Okay, that means this AC element will be acting as an autonomous transposon and will be producing some kind of transposase enzyme, and this transposase enzyme will be utilized by the DS element. Okay, and this DS element will be inducing the breakages at the chromosomes and causing a lot of insertion mutations on the chromosomes. Okay, so this is what about the AC and DS elements, and this is what she said about this AC and DS elements. Okay, so upon her hypothesis or her observations or her conclusions, it was said that AC elements was capable of autonomous movements. That means they don't require any kind of other elements to move this AC element from one location to another location of the chromosomes. Okay, actually this AC element has their own genes to code for enzymes, transposition enzymes or integrase enzyme, and they can move from one chromosome to another chromosome. That is the autonomous nature. Okay, autonomous movement. Okay, but DS element only moved from one location to another location in the presence of AC element. Okay, so this was concluded by the Barbara McClintock, and she said that wherever the DS element is getting transposed, okay, it can also move out again from that location. Okay, it can also exit out from that location and restore the actual gene function. Okay, so this is what she said. Okay, so we have three different categories, three different considerations. Okay, if you consider in the first case, okay, consider this is the chromosome 9. Okay, so in the first case, consider that there is no AC element on the chromosome 9. Okay, so if you don't have any AC element, then the DS element is inactive. It will not transpose into any location. Okay, so the chromosome will stay intact. Okay, that means the wild type allele will be in the normal form only. Okay, if the AC element is present in the second case, consider there is a second case. Okay, and there is a AC element present on the chromosome in the second case. Okay, so here we have the chromosome 9. Okay, if the AC element is present, it will produce some kind of enzyme called as a transposase enzyme, and this transposase enzyme will cut the DS element, will make the blunt end cuts, okay, and will move the DS element to different locations, okay. Say it as the DS element is moving into the wild type allele region, okay. So it is inducing some breakages at the wild type allele regions, and it is getting inserted into that regions, okay. So this kind of chromosomal breakages and this kind of movement will change the phenotypic effects of this corn kernels, okay, obviously inducing a lot of mutations in the corn kernels, okay. So that causes the variations in the color patterns of this corn kernels, okay. So this is what they do. Chromosome breaks and the fragment is lost, okay. This is the small fragment that is getting lost from the chromosome 9, okay. So this W expression ceases, okay. The wild type allele expression now ceases, okay, because of the presence of this DS element, okay. So this is about the second case where the DS element is got inserted into different location, causing the chromosomal breakages, okay. And there is also another case where the DS element can exit out itself from that location, okay. So here the DS element is present inside the an allele, okay, inside a gene, okay. And it also has an ability, if AC element is present in that chromosome also, then this AC element will be involving the transposition of this DS element or the exit of this DS element from that location into different locations, okay. So this is what we see in the maize plants or the corn kernel plants, okay. So the DS element can move from one location to another location on the same chromosome, or from one chromosome to another chromosomes, okay. So that this mobilization mainly happens in the presence of AC elements, okay. So AC elements will be the autonomous transposons, and DS elements will be the non-autonomous transposons, okay. So this is about the AC and DS elements, and here we have the color patterns, okay. So genotype small c or the homozygous recessive condition, okay. So they represent the no transposition activities happening, and because of the no transposition activity, since the condition is in the recessive form, you will be having a yellow to white corn kernel, okay. If there is a transposition, consider if you have a transposition in the heterozygous condition where the capital C is getting converted into CT form, that is a transposed form, okay, and the actual capital allele function is getting disrupted, so you will be having a transparent yellow kernel, okay. So yellow kernel which will be in the transparent form, okay. So this is actually this should not be an yellow kernel, this is actually a colorless kernel, okay. This should be a colorless kernel, okay, because the gene which is responsible for the production of yellow color is actually getting disrupted, okay. So because of that thing, the colorless kernels will be produced, okay. And then we have a second case where the heterozygous conditions will be producing for the purple color kernel, okay. So because of if they are present intact in the heterozygous dominant condition, they will produce a purple color kernel, okay. If there is a insertion, this CT, because of the insertion, this because of this DS element insertion, it will again get converted into what? Yellow color corn kernel, okay. So the recessive is involved in the production of the yellow color kernel, okay, and the heterozygous dominant condition is involved in the production of the purple kernel, while if there is any transposition happening in the chromosome, and if the DS element is getting inserted into the dominant allele of this purple color pigment gene, okay, so then the pigment get disrupted and we will produce the colorless kernel, okay, or you can also call that as a transparent corn kernel, okay. And there is also another case where the mosaic pattern also seen, where if the the transposed DS element, if it get exited out of that locations, okay. Here we have the DS element which is which has got inserted into the capital C allele, okay. So if it get exited out from this location, okay, it will restore the normal gene activity. When it restores the normal gene activity, again you will be getting the normal color patterns, okay. So this is what the LA transposition, where you will be having this purple color, and this is after the late transposition, okay, after the transposition, where you will be having the yellow color or the transparent colors, okay. So this type of variation corn kernel variegation in the color patterns of the corn kernels will be will be done by these DS elements only. Okay, so when the DS elements gets exited out of the gene location, okay, when it goes out of the gene locations, okay, so like this into different locations, okay, so it will produce some kind of variegated color pattern in the corn kernels. Okay, that means some part of the seed will be in the purple color and some part of the seed will be in the yellow color or the transparent color. Okay, so this is what about the AC and DS elements. Okay, so here in the first case transposition is not happening, and in the second case also transposition is not happening. Okay, and the heterozygous dominant condition is responsible for the production of purple color kernel. Okay, and in the third case we will be seeing the transposition. Okay, so when the transposition happens, it will disrupt the gene activity and convert the purple color pigment coding gene to colorless pigment coding gene. Okay, so it will code for the colorless corn kernel. Okay, and it can also result in the mosaic transposition, mosaic development of this corn kernel. That means variegated corn kernel will be produced when the DS element, which is actually present in the gene location, which is actually disrupting the gene activity from producing the color. Okay, when it moves out of the gene location, it will restore the gene activity. This will restore the gene activity, and now the gene activity will encode for the purple color. Okay, so this growing corn kernel will have two types of colors. Okay, so one is during the early stages, that is the the yellow color, and one is the after the transposition activities, that is the purple color. Okay, so this is the purple color is mainly due to the restoration of this gene activity when the DS element has got exited out of the location. Okay, so this is about the DS element and the AC elements. Okay, then we have the P elements in Drosophila. These P elements are majorly responsible for the hybridogenesis. Okay, that is production of a sterile hybrid. When you cross a female Drosophila, female laboratory strain of a Drosophila with the male wild type Drosophila strain. Okay, so when you cross these two Drosophilas, you will be getting a sterile hybrids. Okay, why it happens? Because this P element will be performing the robust transposition activities inside the female Drosophilus, and causes the inactivation of certain genes which are involved in the fertilization process. Okay, now if you look at the structure of this P element, it includes the 31 base pair inverted repeats, terminal inverted repeats. If you consider this as a long DNA sequence as a P element, okay, at the terminal ends, you will be having this 31 base pair inverted repeats. Okay, which we also call as a TIR, terminal inverted repeats. And after this terminal inverted repeats, you will be seeing internal inverted repeats, which are of 100 base pairs, sorry, 11 base pairs. Okay, these are also called as IIRs. Okay, these are called as internal inverted repeats, and which will be involving in the binding of the transposase enzyme. Okay, so they will be acting as sites for the binding sites for the transposase enzyme. Okay, and these two are separated by some kind of a spacer regions. Okay, here we have the spacer regions over here. The length of the spacer region is about 100 base pairs. Okay, so this spacer region separates the terminal inverted repeats and the internal inverted repeats, and internal inverted repeats will be acting as a binding sites for the transposase enzyme. Okay, so if you look at the structure of this transposase enzyme, this enzyme contains certain motifs which forms a domain called as a tab domain. Okay, so this tab domain is specific for this P elements only, and this is actually a DNA binding domain, and this binding domain is in beta alpha beta form. Okay, that means so beta alpha beta secondary structure will be involving in formation of this tab domain. Okay, so this kind of super secondary structure involves different types of motifs. So one motif is what? The C2CH zinc coordinating motif. Okay, so this C2CH zinc coordinating motif contains two cytosines separated by different types of amino acids, and again you will be having a cytosine, and then you will be having the histidine molecule. Okay, which are coordinated by the zinc atom, divalent cation. Okay, so this kind of motif makes the tab domain. Okay, so this tab domain will be acting as a DNA binding domain for the P element. Okay, so the in the transposase enzyme. Okay, now an event called hybridogenesis. Okay, where the P element is responsible for production of a stellar hybrid. Okay, So when you cross the females from a laboratory strains of D. melanogaster or Drosophila melanogaster with the males of wild populations, the offspring which you get in the F1 generation will be the sterile offspring. Okay, this is due to lot of genetic malfunctions or the chromosomal abnormalities. Okay, where the P elements or because of their robust transposition activities, they cause a lot of insertions and the chromosomal abnormalities in the offspring. Okay, so that is what happens. Okay, so the explanation about explanation for the hybrid degenesis is given as genomes of wild fruit flies contains inactive versions of P elements. Okay, that means the P elements which are found in the wild fruit flies, that is the male fruit flies, are inactive, while the typical DNA transposons comprising the transposer gene flanked by inverted terminal repeats, but the laboratory strains lack these elements. Okay, so the laboratory strains, what they have is they don't have these P elements at all. Okay, understood? So this because of the presence of this inactive versions of the P elements in the male wild populations and lack of P elements in the laboratory strains of these female species, and when you cross this both females and the males, okay, the resulting male will get some kind of some kind of P elements, okay, active P elements in them. Okay, so these active P elements in the offspring will cause the chromosomal abnormalities or the genetic malfunctions in the sterile in the hybrid, and will make the hybrid to be sterile. Okay, will make the hybrid sterile. Okay, so that is what happens. Okay, so how this the activation occurs, like why the P elements which are in the inactive form in the male fruit flies or the wild fruit flies getting activated when it is getting into the females flies. Okay, so when the sperms of this male fruit flies get passed on to this female fruit flies, okay, when they intermix. Okay, so this male fruit flies will transfer the P elements into the male fruit flies will transfer the P elements into the female fruit flies. Okay, and that causes the activation. Okay, and this activation is exactly not known, but most of the scientists believe that the activation of these P elements in the Drosophila mainly due to the tissue specific alternative splicing. Okay, that means introns will be spliced in a different way, which involves removal of a certain specific type of introns, and which produces the coding sequences, and this coding sequence is mainly responsible for the production of a transposase enzyme, and this transposase enzyme will activate the P elements, and this P elements now undergoes the transposition activities to cause the chromosomal abnormalities or the genetic malfunctions. Okay, and this is what the structure of this P element. Okay, so what we have here is the exon 1, that is the coding sequences, and exon 2, exon 3, and exon 4. Okay, 4 exons, we have the 4 exons. Okay, and we also have intervening sequences over here, the introns in intervening sequence 1, IVS2, or intervening sequence 2, and IVS3 or intron 3. Okay, so these are the 3 introns which are present in between these exons. Okay, we also have the transposon binding site, the 10 base per transposon binding sites, or the internal inverted repeats. Okay, these are the internal inverted repeats. Okay, and we also have the terminal inverted repeats over here in these locations. Okay, and here also we have the 10 base pair internal repeats. Okay, binding or transposon binding sites. Okay, so the alternative splicing produces one kind of mRNA in the germline cells. Okay, so that means this mRNA will be containing no introns. Okay, only exons will be present. Okay, so this germline mRNA will be containing only exons, no introns will be seen. Okay, but in the case of somatic mRNA, which produces the 66 kilodalton repressor, okay, which represses the activity of these P elements, okay, which suppresses the activity of these P elements, okay, contains one intron. Okay, or the splicing is on the alternative way. Okay, so it doesn't contain the exon, what you call that, yeah, exon 4. Okay, so it doesn't contain exon 4. Okay, so due to the absence of this exon 4, it will produce the repressor molecule. Okay, so the transposase enzyme will now act as a repressor molecule. Okay, and then you also have a different type of mRNA that is called the KP mRNA, which only contains one enzyme. Okay, so this is actually present in the female flies. Okay, so this is about the structure of this transposable element, P element. Okay, and this is how the alternative splicing will lead to the production of the actual transposase enzyme. Okay, so this transposase enzyme is 87 kilodalton weight, and this is the actual element. Okay, so this 66 kilodalton transposase enzyme will be acting as a repressor molecule and it represents P elements. Okay, but this one 87 dalton transposase enzyme will mobilize the P elements from one location to another location in the hybrid. Okay, so based on the hybridigenesis, there are two systems. Okay, they have categorized two systems. Okay, in first, flies divided into inducers and the reactors or reactives. Okay, I strains and the R strains. Okay, so when you cross this I males, inducer males, okay, with the reactive females, okay, hybridigenesis was observed. Okay, so when I males were crossed with the R females, hybridigenesis was observed. Okay, but in the cross of of R females, I females, okay, with R males, okay, so there was no hybridigenesis observed. Okay, that means the reverse conditions will not be giving any kind of reduced fertility. Only the inducer males when crossed with the reactive females resulted in the hybrid digenesis. Okay, and in the second system which is responsible for the hybrid digenesis. Okay, so in this system the flies were categorized into P strains and the M strains. Okay, P strains responsible for the paternal characters, okay, which contributes the paternal characters, and the M strain contributes the maternal characters. Okay, and when you cross the P strains male with the M strain female, now this will cause the hybridigenicity, or this will produce the sterile offspring. Okay, and when you cross the paternal contributing female with the maternal contributing male, okay, so this will produce the normal offspring. Okay, here we will be having the normal offspring. Okay, so these are the two different systems responsible for the hybridigenesis. Most of the scientists have classified this hybridigenesis into two systems. Okay, so in the first systems, they have placed this inducers and the reactive species, and reactive strains, and in the second system they have classified them as the P strains and the M strains. Okay, now, so the dependence of the hybridigenesis on the origin of female in a cross shows the cytoplasm is also an important. Okay, so because the cytoplasm produces the P factors. Okay, so these P factors will be acting as the repressor molecules. Okay, the lines or the strains of flies contain the P elements regarded as the P cytotop, while the strains which doesn't lack lack the P elements are considered as the M cytotop. Okay, so this P cytotype strains or the lines, okay, contains the P elements. Okay, while the M cytotop doesn't contains any kind of P elements. Okay, strains that lack P elements capable of producing a 66 kilodalton proteins, that is actually a transposase enzyme, but this transposase enzyme is not in the active form. Okay, so this is in the some other malfunctional form. Okay, that abnormal function gives a repressor activity. Okay, so this 66 kilodalton repressor will be binding to the P elements and will be repressing the activity of the P elements. Okay, so the strain flies that lack P elements capable of producing a 66 kilodalton protein, but they also exhibit the P cytotop conditions. Okay, that means they can also be considered as a P cytotops. Okay, parental contributing cytotops. Okay, now as I've said when you cross this maternal contributing male or the M cytotype male with the paternal contributing female or the P cytotop female, okay, so this will result in the fertile offspring. Okay, and when you cross this paternal contributing male or the P cytotype male with the maternal contributing female or the M cytotype female, this will result in the hybridigenesis. Okay, this is due to transpose of this is due to transfer of this P elements from the male to female. Okay, and in the female, this transposable elements or the P elements will get activated. Okay, because they produce the transposase enzyme due to this alternative splicing activities, and this P elements will now undergo rapid transposition activities, causing the chromosomal abnormalities or the genetic malfunctions in the offspring. Okay, so that is what happens. Okay, p elements mobilize the p elements mobilize when exposed to cytoplasm lacking the repressor molecule. Okay, sorry, the cytoplasm which lacks the repressor molecules, in the M female. Okay, the M cytota female. Okay, it lacks the repressor molecule. Okay, whenever the p elements get mobilized into the cytoplasm, they get activated. Okay, the burst of transposition events inactivates the genome by random insertions. Okay, I've said you right. So whenever it get mobilized into this cytoplasm which is lacking the repressor molecules. Okay, so this will produce the actual transposase enzyme. P element will now produce the transposase enzyme, and this transposase enzyme will involved in the burst of transposition events, and this heavy transposition events will cause the insertion mutations. Okay, random insertion mutations, and inactivate the entire genome. Okay, that will be resulting in the sterile female. Okay, so this is about the p elements which are found in the Drosophila. Okay, so this diagram actually tells you how this hybrid degenesis getting formed in this offspring. Okay, so if you have this p minus and the p plus strains, okay, p minus indicates the normal male or the M cytotype male, and p plus indicates the p cytotype male. Okay, that is the p element containing male. Okay, so female. Okay, so when you cross these two, the repressor molecules will be present in this female cytoplasm. Okay, since the repressor molecules are present in the female egg cell or the cytoplasm. Okay, this repressor molecule will inhibit the p elements, that are those which are coming from the male strains. Okay, so this will cause the, when they undergo the fertilization, repressor molecule will stay intact inside the zygote. Okay, these are the repressor molecules. Okay, and this repressor molecule will continuously inhibit the p element, so that will be your F1 generation fly will be in the fertile form. Okay, but in the case of this p cytotype male and the M cytotype female, okay, where the p element inactive p elements are present in this male, okay, and the p elements are absent in this female. Okay, so when they involved in the fertilization process, okay, so this p elements will get mobilized into this egg cell during the fertilizations. Okay, and in the zygotes, repressor molecules are not present, where only the transposition elements are present, because this kind of mobilization will now induce the alternative splicing and produce the transposition enzyme in the zygotes. Okay, so when it produces the transposition enzyme in the zygotes, they will cause the random insertion events or the mutations in the hybrid, and that will lead to the sterile fly. Okay, so that will lead to the production of the sterile fly. Okay, so this is about the p element and its role in the hybrid degenesis. Okay, and this is actually the transposition mechanism which is involved in transposition of this p elements. Okay, first it makes the staggered curves. So the transposition enzyme makes the staggered curves in the donor molecule. Okay, and it will mobilize the p element into the target sites. Okay, here, and it will insert them into the target sites. Okay, and these target sites actually these target sites were also cut by this transposition enzyme only in the as equivalents. Okay, or this transposition will make the blunt ends in the targets recipient sequences. Okay, and staggered curves are made on the donor molecule. Okay, and here what we have is the recipient. Okay, on the recipient, we transposition makes the blunt ends. Okay, and the staggered cuts on the donor molecule. Okay, so this is how the p element get transposed into different locations. Okay, and then we have the retrotransposons. Okay, retrotransposons are actually classified into LTRs and non-LTRs. Okay, there are also two different categories. Okay, autonomous LTRs and also the non-autonomous LTRs. Okay, so LTRs are retrotransposon which contains long terminal repeats. Okay, at terminal ends. Okay, terminal ends of any retrotransposons. Okay, so there is a long repeat. Okay, which is actually bigger than the actual transposable element. Okay, so this kind of long repeat at the terminal ends. Okay, which are also called as the LTRs. Okay, makes a major difference between the LTR retrotransposons and the non-LTR retrotransposons. Okay, so LTR transposons, we have TY element in the east. Okay, Saccharomyces cerevisiae contains the transpose element TY, which are the LTR retrotransposons, and the copia elements in the Drosophila melanogaster, which are responsible for the apricot color, and TNT1 LTR retrotransposon N tabacum. Okay, so this species contains the LTR transposons. Okay, the same kind of non-LTR transposons which are regarded as the retrotransposons, which doesn't contain any kind of long term repeats. Okay, so these are present in the L1 sequences in the humans. Okay, so these L1 sequences are majorly belongs to the line families. Okay, long interspersed nuclear element categories. Okay, and we also have the B1, which is found in the mouse. Okay, B1, B2, 1D, and B4 also present in the mouse. These are non-LTR retrotransposons. Okay, and SYN4 is present in the Z maze. Okay, Z maze or plant in this plant species, the C14 will be presenting. And we also have the signs, which is actually non-LTR transposons. Okay, so all the ALU elements belong to this sign categories. Okay, in mammals, the pseudogenes of Pol3 transcripts. Okay, these will be the signs, so short interpersonal nuclear element categories, which are also non-LTR transposons, retrotransposons. Okay, and if you look at the terminus or the ends of this transposons, long terminal repeats are present in the LTR retrotransposons. No repeats are present. No repeats will be present in the non-LTR of the signs. Okay, and the target repeats should be around 4 to 6 base pair length. Okay, if the retrotransposon want to get into the different location on the chromosome. Okay, that different location should contain 4 to 6 base pair length target repeat. Okay, so this 4 to 6 base pair repeat will allow the retrotransposon to get into that location. Okay, and the 7 to 21 base pair target repeat should be 7 to 21 base pair target repeat is required by the non-LTR transposons. Okay, the same thing goes for the signs also, because it is also a non-LTR transposon. The enzymes involved here is ribotranscriptase or the integrase. Okay, and for the non-LTR transposons, endonuclease is required to cut the sequences in the medial. Okay, and organizations may contain introns, those retrotransposons. Okay, which are removed during the transcription process, after the transcription process. Okay, and 1 or 2 uninterrupted ORFs. Okay, operating frames, which produces which doesn't code for any kind of proteins, but they are just regarded as the ORFs, operating frames, or the gene sequences. Okay, and this also this doesn't science doesn't contain any kind of introns. Okay, so lines which are present in the non-LTR retrotransposon category, they are referred to as autonomous retrotransposons, because they can produce their own transposase enzyme. Okay, reverse transcriptase enzyme and also the endonuclease enzyme, while the signs they are quite dependent on the line elements. Okay, line transposons. Okay, so this is major differences between the LTRs and non-LTRs. Okay, so the categorization also given here. All autonomous LTR transposons, retrotransposons, they have same structure. Okay, they contain same kind of genes which encode for same kind of proteins. Okay, even if you talk about the copia elements which are found in the Drosophila, and TY1 element which are found in the this Saccharomyces cerevisiae or yeast. Okay, so these two are different transposable elements, but when you look at the structure, they both contain same structure. Okay, they have a 3' long terminal repeats and a 5' long terminal repeats. Okay, and which are separated by the some spacer sequences. They also have the GAG gene. GAG in the sense the group antigen specific genes. Okay, group specific antigen genes. Okay, so this GAG codes for the viral proteins. Okay, so they also have the PR gene, integrase gene, reverse transcriptase genes, and RNase H gene. Okay, so all these genes are transcribed by the polymerase enzyme, RNA polymerase enzymes. Okay, so the same structure is present in the copia as well as the TY1. Okay, and the same goes for the gypsy and TY3 also, which are also retrotransposons. Okay, and which are also found in the this yeast and Drosophila, and they also contain the same kind of gene sequences. Okay, the structure is same for all non autonomous LTR transposons. Okay, we also have a non-autonomous LTR transposons which are mainly dependent on the autonomous ones. Okay, so they require some kind of enzymes from the autonomous LTR transposon for their transposition property, for their transposition mechanisms. Okay, so major non-autonomous LTR transposons are LARD and TRIM. Okay, so I'll explain about this also. And then we have the non-LTR transposons, signs and lines. I have already discussed this signs and lines in the repetitive and interspersed DNA sequences. Okay, so you can check out that chapter. The major question is, how these retrotransposons can able to transpose from one location to another locations. Okay, so because they have this gag genes, Paul genes, and envelope genes. Okay, they code for an mRNA transcript. Okay, this mRNA transcript is spliced into different this is spliced in different ways. Okay, producing different types of proteins. Okay, the gag proteins and Paul proteins and envelope proteins. Okay, after the transcription, it will get spliced into different mRNAs. Okay, when this different mRNAs undergoes translation, produce different set of proteins. Okay, so one is a gag protein and one is the Paul protein, and one is the envelope proteins. Okay, and the splicing of this different mRNAs also produces different kinds of proteins. Okay, subgroup proteins. Okay, so we have gag related proteins, that is MA, CA, and the NC proteins. Okay, so MA is responsible for the matrix of creation of the matrix. Okay, and CA is responsible for the production of this capsid layer, and NC is responsible for the nucleocapsid. Okay, so packing the entire RNA molecule inside a protein coat. Okay, so that is called as a nucleocapsid. Okay, so these proteins are responsible for the formation of this nucleocapsids. Okay, then we have the PR proteins, which has the protease activity, and the RT proteins, which has a revert transcriptase activity, and we also have the IN, which will be involved in the integrase activities. Okay, which integrates the transposons into different locations. Okay, and then we have the envelope proteins subgroup contains the SU and 3M. SU is a surface protein which provides spikes to that molecule, or the viral-like protein molecule will have spikes. Okay, the spikes are majorly found from this SU proteins of the surface protein. And then we have the TM, transmembrane proteins. Okay, which mediates the virus and host fusions. Okay, so these are the different proteins which are produced from single mRNA transcript that majorly comes from the retrotransposons. Okay, how does this retrotransposition takes place inside the human genomes, or the eukaryotic genomes? Okay, first what happens is a retrotransposon is transcribed from the RNA polymerase 2 from a promoter which is present in the 5' long terminal repeat region. Okay, when it gets transcribed, okay, it produces a mRNA molecule, and this mRNA molecule is translated in the cytosol to give the proteins. Okay, and these proteins are of different types: gag proteins, envelope proteins, and the Paul proteins. Okay, so these gag structural proteins, okay, which are the first major proteins of this mRNA transcript. Okay, so these gag structural proteins form a virus-like particles. Okay, not actually a virus, but it looks like a virus. Okay, which also contain the spikes. Okay, spike glycoproteins on which has a nucleocapsid, and all the things of a virus. Okay, will be present in this virus-like particles. Okay, and the major protein responsible for the formation of this virus-like particle is the gag structural proteins. Okay, when this virus-like particle is formed, okay, so this will make the RNA to get transcribed into to get reversely transcribed into DNA molecule. Okay, or the double-stranded DNA. Okay, so first it produces a RNA DNA hybrid. Okay, so this RNA DNA hybrid later gets cleaved by an RNA is H enzyme. Okay, so into RNA and the cDNA. So this cDNA will now goes and gets integrated into different locations. Okay, and there at that location a retrotransposon gets added. Okay, so making the complete retrotransposons. Okay, so by doing this, the original retrotransposon is also conserved, and the extra retrotransposon is also formed at different locations of the chromosomes. Okay, so this is what the transposition mechanism which is involved in the retrotransposons. Okay, and then we have the copia elements in the Drosophila. So these copia elements which are present in the Drosophila were discovered by the David Hogness and his colleagues. Okay, David Finnegan and Gerald Rubin and Michael Yem. Okay, so they discovered this copia elements in the Drosophila melanogaster. Okay, so why they are regarded as the copia elements? Because they are transcribed in copious amounts of the RNA molecules. Okay, because of that production of RNA amounts in the copious amounts, they are regarded as the copia elements. Okay, and these copia elements are present in 10 to 100 copies in the genomes of Drosophila cells. Okay, and mapping studies show that they are transposable to different chromosomal locations and are dispersed throughout the genome, because they are regarded as the interspersed elements, okay. Same as the transposable elements only, okay. And each copia element contains an approximately 5,000 to 8,000 base pair of DNA, which includes the long terminal repeats. You can also call it the long direct terminal repeats, DTR sequences, okay. And these long direct terminal repeats are of 267 base pair length, okay. And within each DTR there is an inverted terminal repeat, okay, ITR, which is of 17 base pair length, okay. So these short ITR sequences are characteristic features of the copia elements, okay. So these DTR sequences are also found in other transposons of other organisms, okay, but they are not universal, okay. So these DTR sequences are not present in most of the organisms, but some organisms contain these DTR sequences, okay. And the very unique thing about the copia elements is they contain ITR sequences in between the DTR sequences, okay. We have a direct terminal repeats over here, which are inverted, and between these inverted sequences we have a inverted terminal repeats of 17 base pair length, okay. So and two such DTR repeats are present, in either ends of the transposable elements, okay. The entire length of this copia gene is around 5,000 base pairs, okay. And this copia like elements, they are responsible for the eye color mutations, okay, production of white apricot, okay. So what is the big thing about this copia elements, okay. See, insertion of a copia is dependent on the presence of ITR sequences, okay. If the ITR sequences are present, then only the insertion will be made possible, okay, because transposase enzyme will cleave the ITR sequences only, not the DTR sequences, okay. And the copia like element demonstrate regulatory effects at the point of their insertions in the chromosomes, okay. Whenever they get inserted into different locations, the promoter elements which are present in the copia elements, they regulate the gene activities of the neighboring genes, okay. And certain mutations, including those affecting eye color and segment formation, are due to copia element insertion within the genes, okay. They also involved in the change of eye color in the Drosophila species, okay. For example, the eye color mutation white apricot is caused by an allele of white gene which contains copia element within the gene, okay. Transposition of the copia element out of the white apricot allele can restore the wild type allele, okay. So that is what we discussed, right? This kind of a DNA transposition can get into the one location, or it can also get exited of that location. So same goes for the retrotransposons also. So these copia elements can also move from one location to another locations, and can also get exit from that location, okay. When they get exited of that location, that will result in the restoration of the normal type allele or the wild type allele function, okay. So that is what I meant. Like when this copia element is present, along with this white gene, the allele which codes for the white gene, okay. So that will be disrupting the gene activity, or the mutating the gene activity, and causing the eye color to get mutated, okay. So the eye color mutation white apricot, okay. And this is what seen in the case of copia elements, okay. Copia elements also contains GAG, PR, IN, RT and RH genes, okay. So which code for different proteins, okay. So the same the reverse transcriptase mechanism which I have discussed in the previous slide, okay. So first it produces an RNA intermediate, then it gets translated into different proteins, okay. And the GAG proteins form the virus-like particle structure, okay. And in this structure, the RNA will get converted into the DNA molecule or the RNA-DNA hybrid, and it get cleaved by this RNase gene enzyme, okay. That is RNase H, okay. So when it get cleaved by the RNase H, the only DNA molecule will now get inserted into different locations, okay. And there the new retrotransposon will be produced, okay. So this is what the copia elements does in the the Drosophila melanogaster, okay. The same thing, every retrotransposon perform their transposition activities in the same way, okay, by involving a RNA as an intermediate, okay. Then we have the LAD, which is also called as the large retrotransposon derivatives, which are derivatives of the actual retrotransposon, but doesn't contain any kind of autonomous properties, okay. They don't produce any kind of transposase genes, okay, transposase enzymes, okay, or the reverse transcriptase enzymes, or the any kind of endonucleases, okay. So they lack long terminal repeats at both ends, okay. So because of that, they don't have autonomous properties, okay, and they require autonomous transposons, autonomous LTRs to be involved to be involved for their transposition mechanisms. Okay, involved in the transposition mechanism for this large transpositions. Okay, so since they do not encode the proteins for their own movement, they rely on the machinery of these related autonomous elements for transposition. And LARD can also influence the genome structure and function by inserting themselves into new locations, and they also disrupt the genes, or they can also regulate neighboring genes, okay, when they get inserted into different locations. Okay, and they also contribute genetic diversity and evolution by promoting recombination and also genomic rearrangements. Okay? Lot of recombination events are also promoted by these large sequences. Okay, they also act as a regulatory elements. Okay, then we have the trim transposons or the terminal repeat transposons. Okay, in miniature. Okay, so these are called as a terminal repeat transposons in miniature, which are also derivative of this autonomous transposons. Okay, so they also depend on the transposase enzyme on other proteins which are encoded by the autonomous elements. Okay, for their movement. Okay, for the transposition mechanisms. Okay, like TARD, okay, TRIMs also can insert into various genome locations, okay, affecting the gene functions and also regulations. Okay, all the transposons, whenever they get into any different locations, they regulate the gene activity. Okay, they regulate the gene activity of the neighboring genes. Okay, they disrupt the they either disrupt the gene activity, or they either cause the mutations. Okay, they either cause the insertion deletions on the inversions at that sites. Okay, a lot of things will happen. Okay, they regulate the lot of gene expressions the neighboring genes. Okay, and they also play a role in genome evolution by facilitating genetic variations and the rearrangement. Okay, same thing which is done by this LARD. Okay, so the how this LARD is involved in the genomic recombinations or the genetic rearrangement. The same way TRIM is also involved in the rearrangements of the genes or recombinations of certain genes. Okay, and TRIM can also serve as regulatory elements, modulating expression of the adjacent genes. I've said you right. A lot of transposons will be acting as a regulatory elements only. They regulate the action of expression patterns of neighboring genes. Okay? So this is about the retrotransposons. Okay, so one kind of mutation which is seen in this grapes. Okay, so that is due to the retrotransposons. Okay, there is a gene called as WVMYBA1. So this gene is responsible for the production of purple color grapes. Okay, so when it get mutated, or when a retrotransposon is called as a GRET1 retrotransposon, when it comes and gets inserted into this neighboring regions of this gene. Okay, so this gene is present on a chromosome. Consider this is the chromosome number 7. Okay, at the beside this gene, if you insert a GRET1 retrotransposon, okay, this will alter the expression of this purple pigment producing gene, and it will make the gene to be in the inactive state. Okay, or it will result in the low production of the anthocyanin, which will cause the grapes to look as in white colors. Okay, this will result in the yellow color or white color grapes. Okay, and if the mutation is reversed, okay, when the retrotransposon gets exited out of this site, okay, and when it gets exited out of the site, it will leave some some kind of some part of this retrotransposons, okay, which will also regulate the activity of this WVMYBA1 gene, okay, which is involved in the purple color anthocyanin production. Okay, anthocyanin pigment production. Okay, so when it regulates, it will cause the gene to produce a red color pigment. Okay, so this kind of variation in the phenotypic patterns of different colors. Okay, so because of these retrotransposons, okay, these are considered as the gene regulatory elements. Okay, they are majorly considered the gene regulatory elements because they regulate the expression patterns of these genes. Okay, so since this WVMYBA1 gene is producing one kind of protein when it is present in the wild form. Okay, when there is a transposon present behind this gene region, it will be involving in the low production of the anthocyanin or the disruptor synthesis of the anthocyanin production. Okay, that will result in the production of yellow color or white color grapes. Okay, and if there is some part of this retrotransposon behind this before this WMYBA1 gene. Okay, so this small part also regulates the activity of this gene and causes the grapes to be in the red color. Okay, so this kind of regulation thing is also done by the lot of transposons. Okay, majorly transposon will be acting as a regulatory elements only. Okay, so they also cause the mutation, they also cause inversion deletions, all these things. Okay, so this is about the transposons of eukaryotes. Okay,